Welcome to the Cell Collective, a community web-based platform for modeling biological processes. This platform has been created to allow laboratory scientists to model biological processes in a highly collaborative and intuitive fashion. You no longer have to be a computer scientist or a mathematician to start taking advantage of some of the powers of computational systems biology. I am Tomáš Helikar, one of the creators of the Cell Collective, and I will be taking you through a brief tour of the platform. This is the first introductory part of our tutorial series, with subsequent episodes coming in the near future. In this tutorial, I will demonstrate how different features in the Cell Collective can be used, and most importantly, how they are directly connected to laboratory experiments. The first page you will see once you're logged in is the home page, which will contain various notifications about the models you're working on, experiments you perform, as well as any collaborative work that you take part in. All models can be accessed in the Models component from the top menu. There are several types of models that can be created in the platform based on their accessibility and visibility. We will focus on the published models that are visible and available to all registered users. Our group's research centers around complex cellular signal transduction networks. In the course of our research, we have constructed and published one of the largest computational models of signal transduction in a fibroblast cell, which is also the first model you can see under Published Models. The Model Biologic button on the Models page takes us to a page that contains more details about the model, including the individual components of the virtual cell. Specifically, the table on the left contains all species that are part of the signal transduction machinery. From here, the regulatory mechanisms of the individual species can be accessed and easily modified. The table on the right lists external species that represent the extracellular environment. In the case of the fibroblast model, these species include various G-protein coupled receptor ligands, as well as external calcium pump, extracellular matrix, epidermal growth factor, and a couple of stress stimuli. As an example, I will explore the regulatory mechanism of RAS, an important regulator of many cellular functions, including cell growth. Each regulatory mechanism is specified as a set of upstream positive and negative regulators, which can be defined in their respective centers. For example, under the positive regulation center, we see that the positive regulators or activators of RAS in this particular model are RAS-GRF, SHP2, and SOS. This is also summarized in plain English words at the top for users' convenience. More complicated relationships within the regulatory mechanisms can be defined in each of the positive regulator centers. For example, let's assume that laboratory data suggests that the activation of RAS by SOS occurs only when RAS is localized in the plasma membrane by the phospholipase PIP245. To add this information into the model, the user would create a condition under the SOS center called localization, and specify PIP245 as part of this condition. Again, users can review the condition in plain English words at the top. All positive regulators and any condition specified for the RAS regulatory mechanism can be reviewed on the main page. The same way positive regulators are specified in the Positive Regulation Center, the user can specify negative regulators in the Negative Regulation Center page. I will now return to the fibroblast model and show how this model can be simulated and analyzed. There are two simulation tools available in the current version of the Cell Collective. Let's first start with the real-time simulations. Here at the top, you can see the external species representing the extracellular environment, whose activity or concentrations can be changed with these sliders in real time to view how the dynamics of the system change in response to the extracellular stimuli. By default, all external species are set to 0, so before the simulation is started, we will change the activity levels of these species to more meaningful values. On the right, there is a legend which contains the species whose dynamics will be visualized during the simulation, as well as a list of all species in the model. I will remove some of the species in the legend placed in there by default, and I will replace them by ones that our group has been interested in. Now we're ready to start the simulation. As you can see, as this chart evolves, the activity levels of most of the species in this graph are low, and that is because these species are specifically ERK and AKT are associated with growth. And as you might have noticed, when I was changing the default values of the external environment, I skipped ECM and EGF and I left them set to 0. I did that on purpose so that I can show you how some of the species in the model will respond when I change the activity levels of EGF and ECM to higher levels. And you can see the response is already happening. In addition to being able to see the changes of activity levels and or concentrations in the cell as a response to changing extracellular environment, we can also perform various mutational studies such as loss or gain of function. In this tutorial, I will show you how we can mutate RAS, and we can simulate gain of function on RAS, and RAS is very important in many diseases such as cancer. And now I am simulating gain of function on RAS, and so we will add RAS as one of the species in the graph, and you can see in purple RAS is climbing higher and higher, and it will eventually reach 100%. And you can also see that the activity levels of all of the other species has been increasing, and specifically in terms of ERK and AKT, to make sure that their activity levels are increasing as a result of the mutation, I will remove ECM and EGF from the extracellular environment, and we can see that the activity levels of ERK and AKT remain still relatively high even in the absence of growth stimuli. Users can also pause and play the simulations at any time. So you can see that the real-time simulation tool is very intuitive and easy to use, and provides a great way to test many biological what-if scenarios. Going back to the main simulation page, I will now demonstrate the dynamic analysis simulation tool. The dynamic analysis tool allows users to conduct in silico experiments that consist of hundreds of simulations, where each simulation of the virtual cell is done in a different extracellular environment. Using these dual sliders, users can specify the range of activity of each external species, and the simulation tool will randomly pick an activity level for each simulation. This way, users can easily and quickly perform analyses and generate things such as dose-response curves. As an example, I will show how the fibroblast model can be analyzed and a dose-response curve involving epidermal growth factor generated. First, I will set the external environment to more meaningful values, just like I did in the real-time simulation. In this case, I will let ECM vary at high levels, and I will leave EGF set to the default values so that we can see the functional response of the various species inside the model as a response to EGF. By default, we can perform 100 simulations, but to save time, I will change that to 10. And we also need to name the experiment. Okay. Now we will run it. You can see that the experiment, the study actually took very little time. And now when the experiment is done, the user can go to the analysis tab where they can generate the dose response curves. And we are now interested in looking at EGF, and for this particular example we'll look at how AKT responds to the various dosages of EGF. Users can also set the Y range of the graph. If they leave it at the default values, the graph will end with the maximum value of the y-axis, or the AKT in this example. Okay, so now we see that as a response to EGF, AKT's activity increases, so there is a positive correlation, as we also know that this is the case from many laboratory studies. The advantage of using this simulation tool is that you don't have to do different experiments to look at different functional dose response curves. So we can now generate a dose response curve for a different species, and so we'll do that for example, ERK and EGF. And as you can see, also there is a positive correlation, just like we would see in the laboratory. And so again, the advantage of this is that we don't have to perform multiple experiments. We just run it once, and then we can look at various dose response curves. Now that we have seen how to access models and how to simulate them, one may wonder where to find the biological information that defines these models. And this is where the knowledge base component of the cell collective comes into play. This is where scientists can track the local interactions between individual biological species as discovered in the laboratory. Because the cell collective is based on a qualitative, parameter-free modeling framework, no kinetic information, no kinetic parameters, which are very difficult to obtain generally, are required to enter into the system or build any models. The main page of the knowledge base contains a list of the species that have been already entered into the system, including the entire model of the signal transduction in a fibroblast cell. Because of its wiki format, all contents in this knowledge base can be easily modified in a very collaborative fashion. Each species in the cell collective has its own page where laboratory scientists can deposit any experimental results that suggest as to how the species is regulated in the context of the given biological process. Each page also contains a reference section where users provide published works to support the information entered into the knowledge base. The final component of the cell collective is the forum, which allows for open discussions regarding, for example, the features of models in the cell collective. This is also a great place to help us make the cell collective better by letting us know about any problems or bugs you may encounter. We are constantly monitoring the forum to ensure prompt responses. And this concludes the overview tutorial on the cell collective, a community platform for modeling biological processes. Thank you and enjoy the software.